Well, first of all, my name is Randy Schekman. I'm an investigator and a professor at the Department of Molecular and Cell Biology at the University of California, Berkeley. I became interested in Parkinson's disease many years ago when my wife first developed the disease. And so I followed it, of course, from a distance. My own research is not directly related to that. And as she grew more ill, I was approached by the chief financial officer of the Sergey Brin Family Foundation. Mr. Brin, the co-founder of Google, has a genetic history of Parkinson's. His mother is ill with the disease. She has a genetic form, a form which Mr. Brin has inherited in part. He's a carrier of a mutation that his mother has 2 copies of. And so because of the nature of the mutation, he is at some risk. He has been very generous in giving support for Parkinson's research to the Michael J. Fox Foundation. But he decided a few years ago that what really is needed is more of an effort at a basic research understanding of where the disease originates, what causes the cells in the brain that make dopamine to cease functioning, to die, and how to detect the disease as it progresses. There are no known biomarkers for the disease. You know, other diseases like heart disease and cancer can be followed with biological measurements. There is no such biological measurement for Parkinson's. It's all based on a physical or a cognitive evaluation, which is gives the impression that disease progresses in very different ways. And that's a problem. It may be more than one disease. We just don't know. We don't know enough about how the biology of the disease. And so Mr. Brin approached me indirectly to lead this effort when my wife died, which was a few years ago. And for the last 2 years, we've been building this network called Aligning Science Across Parkinson's Disease, which is leading to the creation of an international network of investigators. We put out a call for proposals from scientists, physicians, and scientists around the world who we want to come together in the form of teams, at least the core of which these teams should already have been collaborating on some relevant issue. So we want teams that aren't just created for the purpose of getting money. We want teams that have demonstrated the ability to collaborate in a meaningful way, to share their results, to in the give and take of a collaboration, and not in the usual, I'm afraid to say, sort of competitive environment of academic science. And so the reasons for our intention to make this a very open network is that the reward structure in academic science and academic medicine favors the individual and tends to discount the more important roles that are played by collaborators who don't necessarily expect all the credit for the work. They want to share the credit because they want to move the needle forward. So we have developed policies that will ensure that our teams are truly collaborative, that they are willing not only to work within their own team of investigators from different institutions, but within the network of what may be, you know, maybe 100 investigators or so. So we have put out a call. We've received 150 pre-proposals from teams, some of which are from Australia, some from New Zealand, but from all over the world. And we are now evaluating these pre-proposals, and we will call on those teams that seem to be most interesting and most well integrated to write full proposals, which we will then evaluate later this year with the aim of providing funds for the selected teams by summertime. We don't have any preset number of teams that we'll select. Mr. Brin has been very generous. We have a substantial budget. I don't think we'll be able to spend it all, quite frankly. So it's generous, and it's a level of support that is unusual, particularly for basic science. And I emphasize the basic science. Obviously, there's good clinical research going on, but we feel that the clinical research is already well funded, for instance, by the Michael J. Fox Foundation. But what is missing, we feel, and I feel as a basic scientist, is the need for more basic research to understand, even at a molecular level, where the disease goes bad. It may start many, many years before the symptoms arise. There's a very long prodromal phase that can sometimes occupy decades before the movement disorder appears. What is the basis of those curious early symptoms that aren't seemingly related to Parkinson's? How does that inexorably lead to the disease? These are compelling questions for which we need much more insight.
They certainly are very compelling questions. And so where do you see the future frontiers of Parkinson's with the ASAP initiative?
Well, there are three goals really that we have in the initial round of funding. The first, and I think there are three goals that are equally important. The first one is to pursue the known genes. There are now something like 20 different human genes that represent families with a high incidence of Parkinson's, where it is clear that a mutation in a gene predisposes to the disease. It's not, 100% certain, but it predisposes to the disease. What are these genes? What do they code? Those protein molecules that are coded by these genes, which pathways do they intersect? How do they interrelate among these 20 genes? Can one draw from, a kind of a puzzle that put the pieces of the puzzle together to create a coherent picture? Or are there really very distinct pathologies that represent different diseases? We think the genes are one key sort of entree into this puzzle. A second goal is to understand the neuroinflammatory processes that are seen in neurodegenerative diseases, not only Parkinson's, but also Alzheimer's. Are these neuroinflammatory processes linked to the origin of the disease, or do they make the disease worse? Where does the immune system interact at the earliest stages and in the breakdown of dopaminergic neurons? So neuroinflammatory processes and the role of the immune system are another key knowledge point that we wish to establish. A third goal, which we'll set forth in a proposal call later this year, is to simply identify all the neuronal connections that are made between cells that make and secrete dopamine and other cells in the brain. That network is not yet fully established in the human brain. And we think there's a lot of basic neuroscience that is needed to establish what those connections are, because it may be that the primary defect is not in the cells that make dopamine themselves, but rather in the cells with which they interact. Those cells that are involved in producing new dopaminergic neurons, or those cells in the brain to which they connect to send signals that control movement. Those networks need to be more fully established. And an overarching goal that covers all three of these themes is this mysterious prodromal phase, the sometimes 20 years or more between the first kind of curious malady that many people don't often associate with any particular illness, but which often, very often, surprisingly, leads to Parkinson's disease. What is that prodromal phase? How can it be seen? What are the markers, the biological markers that may be discovered that would allow a family at least to chart the path of the disease, to know what their fate is? The most frustrating thing, I'm sure people who listen in will appreciate, the most frustrating thing about the disease is the inability to know where the disease will go next, because it proceeds in many different directions. That was the most frustrating thing to me. I had contact with neurologists all over the world, particularly experts in my wife's, certain form. She had what was eventually diagnosed as diffuse Lewy body disease. I would ask anybody I met who was a specialist in this what to expect next, to describe where she had been, what's going to happen next. I was usually given an answer, and it was always wrong. No one could tell me what to expect. She eventually died, likely of heart failure in the middle of the night. No one told me that that was a possibility. This, of course, is the most frustrating thing for families. And if we can make progress in establishing these biomarkers, then there is a good chance that we can at least advise people how to prepare, what to do, perhaps to forestall what may await a patient with a particular syndrome. So these are our hopes. These are our plans. The work will be basic. We're not going to be supporting drug discovery or clinical, basically work in the clinic. We want to support physicians and PhD scientists who are doing the basic molecular and cellular work. Well, I hope you tune in, because what I have to say I think will chart the path of a very prominent role for basic science going forward to understand where this disease begins and how it progresses, to establish new knowledge that we don't have yet, but which will, if it works, will lead to new therapies.